Good afternoon, everyone. I know you're all very hungry, but this won't be quick. I'll try. Um, in recognition of the continued trend of women in engineering, I would like to acknowledge that this work was conducted by two excellent researchers from my lab, Srishti Johri, she's a PhD student, Roslyn Massey, Dr. Roslyn Massey, a postdoc. She's now at Stanford. And this is a technology that we have gradually worked on and improved over the past almost 5 years now. Starting at the first time we presented it at Fleps at a very simplistic early stage platform. So hopefully this looks a bit more mature. The platform itself is essentially an integrated organic electrolyte gated field effect transistor. So multitude of electrolyte gated transistors integrated with different biorecognition molecules, aptamers, antibodies, nanobodies, perhaps, with a low temperature processed microfluidic channel. And we are currently working on interfacing with both commercial partners and ourselves. So the idea is to get to a point where we can do multi-analyte proteomic analysis, and the target right now is towards neurodegenerative diseases. One of the assets that we have, which allows us to conduct this kind of research, and as we have better equipped ourselves, is the versatility in direct printing. So don't get me wrong, we are very well supported by the conventional microfab, the nanofab for all the characterization, but we also have acquired versatile extrusion inkjet printers and more recently an aerosol jet printer, which has simplified prototyping and allowed us to scale up manufacturing to a point where we are now involved in small-scale biobank studies. We have started clinical trials with local hospitals looking at PET tracer correlation with saliva and serum biomarkers. This is almost a preamble to the extended work that we are aiming to report later this year. And we're based in Ottawa, if I forgot to mention, the capital of Canada, close to Montreal and Toronto, which you probably are more familiar with. So it says non-invasive. I understand it is minimally invasive, but if you understand the domain of neurodegenerative, you would understand why I'm saying it's non-invasive, because the benchmarking in neurodegenerative disease diagnosis tends to be spinal tap or lumbar puncture for CSF, or radioactive tracers for PET, tau. More recently, looking at multi-analyte blood-based analysis. But even then, when it's a mass spec or other criteria, you need a lot of blood. So when you need a lot of blood, you need venous puncture again, which tends to be invasive. So what we are trying to get to is a point where the multi-analyte testing can be done with very small quantities of blood. And we can correlate multiple biomarkers with, let's say, something like capillary blood sampling using a Lancet, something that's already done. The population is familiar with the concept for diabetes management and other continuous health monitoring. In parallel, we're also looking at saliva blood to serum correlation as part of the clinical and biobank studies. And we're constantly seeing if it can be turned more wearable using sweat as or ISF interstitial, dermal interstitial fluid. So the footprint of the device, my job has been made very easy, so I'll breeze through this. We all know organic semiconductors, dielectrics, so we'll quickly jump towards the device configuration. I've highlighted some of the materials we are currently incorporating. So small molecule semiconductor tip sphentosine, with collaborator at U Ottawa, we have started to get really good results integrating single walls carbon nanotubes. Conductors, we continue to rely on metal nanoparticle links, just the resistivity, the low resistivity and superior interfacing. So far, we have not been able to reproduce that with carbon-based inks or PSS variants. Structural materials, we use a lot of hydrogel type materials to control solvent diffusion, limit solvent diffusion, and also to enhance device stability. So we have combinations of PVA, PMMA, PDMS, PCl these three being biodegradable, biocompatible. We are trying to completely step away from PMMA by doing direct immobilization of biorecognition molecules on PCL. Substrates, polyamide, I think it's everywhere. We love it. DuPont gives it us for free. Superior thermal conductive properties, so it is really, in my mind, no replacement as of now for Kapton. And we like that for the flexible aspect too, because if we were to move towards ISF and capillary-based collection of blood, the flexibility will be immensely helpful. For bioreceptors, biorecognition molecules, we have incorporated aptamers and antibodies. And yes, so kind of to point out, so we have reported something in ACS Nano where we have incorporated single-wall CNT for neurodegenerative biomarker. Primarily, we work with TIPS, Pentacine, and P3HD. So electrolyte-gated devices are not new. They have become quite the industry benchmark for a variety of applications. Started more with energy storage, but you see with MEM transistors, memory devices, fast switching logic elements, they're being more and more implemented. But the type of electrolytes is where the sensor applications are different than the solid electrolytes. So for polymer composite electrolytes, mostly used in conjunction with conjugated semiconductors for MEM transistor, logic, etc. applications. But a lot of aqueous electrolytic devices, as you can see, and they are configured as electrolyte-gated organic FETs, EGFETs, organic electrochemical transistors, etc. And the domain there is very focused on directly incorporating biological fluids as the electrolyte, and then use that for diagnostics. So we coined the term OEGFET 5 years ago, and we stuck with it because of the key difference between the conventional electrolyte-gated FET and OEGFET. So conventional electrolyte-gated FET benefits from an ionic media or changes in ionic concentration of the media because of the interaction, charge injection from the electrolyte to the semiconductor gate to the electrolyte, which leads to high current, very high transconductance, but it also leads to pseudocapacitance and shelf life issues. So like how long can you use, especially for if you're looking at repeat measurements or continuous monitoring type applications. OEGFETs, we shield the electrolyte. We don't shield it with like really solid high dielectric layer thicknesses, but we shield it for functionality. So for example, I think in the next slide, I have the layer details. We use porous, semi-porous materials to prevent solvents sinking into the semiconductor material during the integration of the microfluidic channel. Same for the antibody mobilization. So there's some chemical activation is required. So we use underlayers to minimize effects on the conductive gate electrode. And this allows us to sort of get the versatility of binding different kind of bioreceptors. No need to go in details here, but aptamers and antibodies, monoclonal antibodies, they're both synthetic receptor molecules or recognition molecules, widely used in sensory applications. And we have used and compared both of them in our platform. So current targets, alpha-synuclein, amyloid beta, tau protein. We're reporting on all three. In this work, we will look primarily just at the beta amyloid. If you're not familiar, it's one of the key biomarkers that's being investigated for blood-based or fluidic biomarker-based detection, early detection of Alzheimer's disease, which is one of the neurodegenerative diseases that starts with neuronal cell decline, cognitive decline in proper motor functions, and has long-term repercussions, both social and socioeconomic. The challenge is that while the clinical symptoms can take decades to manifest, a lot of the precursors start to show up quite in the early preclinical stage before, let's say, the subject cognitive decline occurs. The challenge is to what concentration can they be reliably correlated? But I think they can. A lot of the scientific community agrees on the topic because currently the alternatives are not scalable. So you can't really do mass screening with something like PET, right? You can't do mass screening with spinal tap. Currently, the only mass screening tool is MiniMoCA, another kind of digital cognitive test. Digital biomarkers have their limitations. Different people have different digital signatures, and there's no way to confirm that what's aging, behavioral versus what could be early dementia and things like that. So adding fluidic biomarkers to the digital biomarkers footprint, as Anna Maria pointed out, making my life easy, is the way to go, and almost entire community agrees on that. So we are trying to do that. And this is a quick preview of how it all started. We had glass, silicon, very simplistic assembly. We reported it at the FLIPS 2019. So that's why we're like, it's poetic to come back to FLIPS and report now in 2024 that we have a really nice sealed system. So how do we fabricate it? As I said, it's scalable to some extent. So we can build on one substrate up to 25 to 50 devices. So every one of these has 5 biosensors on them. Currently, shadow mask CoVAP deposition, so low temperature physical vapor deposition, is what we're using for the source drain electrodes. For the top gate, we have now switched to aerosol jet, which makes life a lot easier because then you can do all of that at room temperature. And this is where most of our functionalization is, on the top gate. And then we assemble it using a chemical adhesion process. And then we test it using our laboratory setup, as well as using a portable reader that a company in Edmonton has developed for us. So very quickly, the early results that we have reported in the latest paper, I wanted to stick to that. In terms of parameter and analysis, we have simplified it because we want to make it portable. Initially, we were doing all kind of complex Marino model, curve fitting, extensive data extraction. Now we are looking at just 3 parameters. So the saturation current level, the midway of the linear region, which correlates to transconductance very well, so capacitance from there. And we're looking at the subthreshold swing to kind of confirm that the threshold, the turn on and on off ratios remain comparable. The sensing mechanism, and this is where it's quite different than the electrolyte gating, right? We don't have ion exchange, we have ion separation. So if you're familiar with the Gibbs-Donnan behavior, when you have a permeable, semi-permeable to somewhat non-permeable membrane at an interface, it separates charges. So charges are not able to travel through it, which creates a capacitance effect. It's a small capacitance effect, but it primarily depends on the binding there, which creates that permeable separation. So for many, it's insignificant. For us, it's the star of the show because it allows us to minimize all the other contributions that's coming from the bulk. So the ionic effects from the bulk doesn't change the sensitive region a lot because all we are looking at is the Debye interaction. Also allows, the Donnan interaction. And it also allows us to some extent overcome the Debye limit that a lot of these biosensors struggle with. Because as soon as you bind large molecules at an interface and you go out of that double layer, Helmholtz Debye double layer, your sensitivity drops. So this is the integrated sensor structure, and during testing, standard transfer functions, IV responses. We have tested over a few hundred devices, so they're quite repeatable performance. In deionized water, obviously no matrix effect, so nice linear, log linear behavior. What is new here, along with the antibody-based biomarker, is we are looking at serum. So we are currently doing a biobank study with serum, but this is a preface to that, where we look at different dilutions of serum. So from the very concentrated serum to 50, 100, 200, up to 1,000, 2,000 times diluted serum sample, and we looked at our sensitivity. And as you can see, for the relevant concentration range, we are in the sensitive region. We do lose sensitivity, and it becomes predominantly matrix effect because of the surface area being saturated and the solution being too inundated with competing HSA and other proteomic markers. But thankfully for us, this is the region of interest where we have been sensitive in serum and saliva, as the testing show. So to summarize, with this work, we've expanded the organic electrolyte gated FET footprint from purely an aptamer sensor to a multi-analyte. So we can incorporate aptamer antibody. We can look at not just metabolomic markers, but large size proteomic biomarkers. We have a broad detection range, which fares really well in the lower concentrations because of the inverse current to concentration correlation. We are seeing high matrix effect in high concentration region, which is something that we are investigating in the current study as well, and see if it's possible to push the detection range a little bit more. And the sensor is stable, laboratory shelf life close to a month. So with that, I'll close, and I'll thank funding partners, collaborators, which there are plenty, to help with this work, and all the excellent researchers in the lab. Thank you.